Welcome to Brain Ponderings, your window into the world of neuroscience research, conversations with experts in their particular area of neuroscience research. Today, it's my pleasure to welcome Grace Stutzmann. She goes by Beth.
Mhm.
Um, and she's been doing a lot of interesting work on suggesting that there's alterations in the way neurons handle calcium, an ion that's very important in their function in aging and Alzheimer's disease. And so, um, welcome, Beth.
Thank you very much. Yes, it's a pleasure to chat with you. And so we've crossed paths before, so good to see you again.
And Beth is a professor and I guess chair
Yep.
of discipline of neuroscience.
Yeah, I can explain if you'd like. We've got research centers and academic disciplines. So basically, if you combine them together, it becomes a standard department.
Okay, and the director for the Center in Neurodegenerative Disease and Therapeutics at the Rosalind Franklin University at Chicago Medical School. Um, Before we get going, I just want to kind of get some basic neurophysiology. So the concentration of calcium ion outside of a neuron is at rest when the neuron isn't firing. It's about 10,000-fold higher
Yep.
than inside the cell. And when a neuron fires, it depolarizes the membrane because sodium channels open, and then calcium channels open. There's voltage-dependent calcium channels, but there's also glutamate receptor
Mhm.
calcium channels. And so that's kind of the framework for this. So the point is calcium is very important
Yes.
for learning and memory, for all the functions of the nervous system. And there's increasing evidence that uh, the inability or problems with properly regulating calcium are contributing to Alzheimer's disease. So, Beth, why don't you talk about your early work in this area? Well, I guess you didn't really start out in you start out in psychology and
Yeah, so I started in more like basic neuroscience,
oh,
neurotransmission, learning and memory
oh,
encoding.
okay.
We don't have to necessarily go back all the way to my graduate studies, but I was always interested in cellular and network mechanisms that went awry that led to brain disease. So my first postdoc was at Yale in the Department of Psychiatry and Pharmacology, but focused more on psychiatric disorders, and looking at individual cells and how they communicated with other neurons to form memories or shape behavior. I then moved to UC Irvine, where that's really where the calcium and the Alzheimer's stories came together. I worked with Frank LaFurla, who's got deep roots in molecular Alzheimer's disease and generating a host of mouse models. And then also Ian Parker, who is a channel biophysicist studying intracellular calcium channels. So it seems like a rather random mix-up, but this is coming around the time where the mutations that will cause familial Alzheimer's disease, such as presenilin and amyloid precursor protein, or APP, are really being discovered and the mechanisms being revealed. And one of the early studies, I think it was out of Echeberry-Gary's group, showing that, family members or people with mutant presenilin, if you took their skin cells and looked at how they functioned, to see what these mutant presenilin genes were doing, they had this increase from their intracellular calcium stores. And, so if you washed on certain agonists, triggered these channels that activated these intracellular calcium channels, they had a much bigger calcium response. And that to me was fascinating because, as a basic, day one of neuroscience school, you're learning how calcium is essential for driving neurotransmitter release, which is what's underlying synaptic transmission. And you alter those properties and you can get memory encoding, you can get changes in synaptic strength. So I'm just quickly, putting these, linking up these dots to form a line to say, wow, if we're seeing altered calcium handling in these familial Alzheimer's, patients' cells, what is that doing to neuronal function? Because you can go from it's controlling everything from gene transcription to, synaptic structure, function, apoptosis. It's just the list is very long. So that is where my starting point was, realizing that most people, or I wasn't coming across anyone in the field really asking those sorts of questions. And it was the memory encoding that I believe most people were, interested in what was going wrong. Like, let's get at that mechanism, then let's try and fix it. And at the time, I guess there weren't a lot of neurophysiologists in the field, that were studying more of the protein biochemistry. So that's really where I took off. And then it gained traction. And the more we keep looking at what is this altered or, abnormal calcium handling doing, each pathway we go down, it seems to be having a very significant effect. So that's the condensed version of how and why I'm still very much committed to this line of research.
So the main circuitry in the brain is there's excitatory neurons that use glutamate as a neurotransmitter, and there's inhibitory neurons that use GABA, and they kind of keep the glutamatergic neurons from firing when they shouldn't be firing. And there is evidence that people with Alzheimer's disease have increased incidence of seizures, clinical seizures, and there's even EEG recordings showing they, even if they don't have seizures, they have kind of hyperexcitability. Then there's some evidence that maybe early in the disease process, the GABAergic neurons may have problems, maybe because they have a high metabolic demand because they fire a lot. And yeah, so we kind of overlapped a lot in when we were working on this, I think. And we had George Martin, Bryce Sofer and his group had made presenilin 1 mutant knock-in mice.
That was my first mouse.
Yeah.
Thank you.
And so he sent them to us. This is when I was at University of Kentucky, and we looked at a whole bunch of stuff. But first, they don't have amyloid in their brain, right? So, and it's because the amino acid sequence of the mouse amyloid beta peptide is different than the human, and the mouse doesn't aggregate. But we did find some calcium handling abnormalities in those mice and actually increased vulnerability in vivo to excitotoxicity. And then we found in cultured neurons that if we expose them to aggregating amyloid beta peptide at a level that doesn't kill the neurons, nevertheless, it makes them more vulnerable to excitotoxicity, and we see like excessive calcium influx. And then you focused a lot on, so we focused a lot actually on calcium influx from outside the cell, and you focused maybe more on, you mentioned these intracellular pools of calcium in the endoplasmic reticulum. So can you kind of go into some of the details of how you do those experiments and what you find?
Absolutely. So I really want to thank you for your seminal work in this field. It was really forming the groundwork for more of the cellular physiology I was interested in. But how we would do these experiments, and this was said largely in Eden Parker's lab, much more the channel biophysicist. So he had a lot of the optical imaging equipment and electrophysiological equipment that I could then modify, and taking mice such as the mutant presenilin that you were just mentioning that you had helped bring to the forefront in the field. I could make acute brain slices from these mouse brains so the tissue is still alive, and I could look and focus in on memory encoding regions. So either say the cortex or the hippocampus. And then using these optical techniques, I could, guide an electrode into an individual, neuron, actually patch onto it. So these electrodes are now contiguous with the cell, and we can kind of, break open a little patch of the membrane in between the tips of the electrode. And then we can also fill these electrodes with really cool things, such as fluorescent indicators that change, that's intensity based on how much calcium is there. So we could fill these neurons with this fluorescent calcium indicator. We could also fill it with compounds that we could trigger that would release calcium from intracellular stores. So it's this caged molecule that once you uncage it, it attaches to its receptor and opens calcium channels. One of them being IP3. So the basic experiment would go, we'd patch onto our electrode or patch onto our cortical or hippocampal neuron, fill it with these fluorescent indicators that would essentially measure calcium. And now we can also control the electrical excitability of the cell. So we could trigger the voltage-gated calcium channels by injecting depolarizing current, right? And we get action potentials, and we'd see the voltage-gated calcium channels open, and all this calcium flood in, and then we can measure the change in fluorescence. So it was great. They're great, really fun experiments. And we could compare that against the, normal mice versus the Alzheimer's mice. And typically we found, and we can do this like in individual dendritic spines, we can do it in the cell body, like wherever we want in the cell, we can focus in on. We typically found that the voltage-gated calcium channel activity was not that much different. But when we uncaged the agonists or the compounds that would release calcium from intracellular stores, right? So there are these pools of calcium being stored within organelles, so the endoplasmic reticulum being one of the biggest ones. We released that calcium store, we saw a massive increase. So we were able to hone in that it seemed to be mostly in these stores. We could also activate those glutamate receptors, so the NMDA receptors, the AMPA receptors, other calcium passing plasma membrane channels. Typically, they weren't that different, although we were seeing increases with NMDA activation, but I'll finish that story in a minute. It was largely these ER stores. And I think you actually had published one of the early papers. It was implicating the ryanodine receptor.
Right.
We'd largely been chasing the IP3 receptor, since that's really where historically the changes were being seen. And so we could trigger the IP3 receptor, see a big response, trigger the ryanodine receptor, also see a really large response, in the Alzheimer's versus the control. And so then it was, well, what is this extra calcium doing, right? How is it regulating neurotransmission? How, neurons communicate in a way that is regulating memory encoding. So we were seeing, again, changes in spontaneous vesicle release. So like how many packets of neurotransmitters were being released was, increased in the Alzheimer's mice, so that could contribute to this baseline hyperexcitability. We were seeing changes or, imbalances in the inhibitory and excitatory tone, right? We can look at, the GABAergic versus the glutamatergic responses, and that was out of whack, so leading to sort of a net increase in hyperexcitability. We could see changes in plasticity. So if we trigger a synaptic response, right, we can stick in a stimulating electrode while recording, look and isolate individual synaptic responses. And those, while they seem to look normal, were actually operating under basically a different system. So if you then normalize this calcium, you see very abnormal changes in synaptic strength. So this was all this excess calcium was very tightly coupled to key mechanisms driving learning and memory encoding. I'm like, okay, now I feel like we're getting somewhere. Like we're understanding the basic nuts and bolts of how and why some of these circuits, essential for memory encoding, are working in this imbalanced, dyshomeostatic state. So those are the types of experiments we started with.
So, yeah, so that's in the absence of amyloid accumulation in plaques.
Correct.
So one big question is, and there is evidence that the presenilin mutations increase the amount of, it's called a long form of the amyloid beta peptide that maybe is more prone to aggregation and accumulation and toxicity. So then the question becomes, which I guess you could have a couple ways. One is that the effect on the intracellular calcium release is upstream, that is, occurs before the amyloid accumulation in the actual people, humans with
Yes.
the disorder. Or it could be the opposite, or more likely, maybe, in my view, there's like a cross-amplifying thing where you get kind of a feed-forward cascade, and once it starts, this snowballs out of control.
That is very much the hypothesis we're working off of, that it's more of a just feed-forward cycle. There's many, many things that could drive this aberrant calcium signaling, but then once it starts, one of the things it drives is one of the secretases, that aberrantly cleaves APP, so you get more of these A beta fragments. It's driving the kinases that are aberrantly phosphorylating tau, leading to increased, phospho-tau, which is also
Yeah.
pathogenic. It's leading to too much calcium being taken up by the mitochondria, so it's spitting out more free radicals, which is running around oxidizing things, including the ryanodine and the IP3 receptors. That's wreaking havoc on, cellular functions. The list just keeps growing.
Yeah, and even in those people, well, and some of the families with some of the mutations, they develop symptoms actually in their late 30s,
Yeah.
but
Mhm.
early 40s. But it's still age related. I mean, they seem fine when they're up and certainly through their 20s. And it turns out that, our brain, it's like our physical performance. Our brain performance starts to decline when we're in our 30s
Oh, yeah.
in some domains. And we can do what we need to do for the most part, and nobody notices it. But anyway, so you mentioned the oxidative stress. So there's a number of like hallmarks of brain aging. One is accumulation of oxidatively damaged proteins, DNA, membranes, inflammation, and uh, what else? Energy metabolism, impaired energy metabolism. And so we did a lot of focus on, oxidative stress at the plasma membrane, impairing some key proteins involved in calcium regulation, like ion motive ATPases, the sodium pump, calcium pump, and also glucose transporter. So what are you interested in or working on any like combining some age-related relevant condition with the mutation or other manipulations of calcium?
So, we are always using aged matched controls in our, if we're using mouse models,
Yeah.
right? So some of these studies are done what we call pre-symptomatically. So cognitively, they seem okay. There's not a lot of histopathology. And then we'll continue looking at changes over time. So as these mice age. Um, so we do see changes in calcium handling in our control, like older control mice. And this is consistent with studies. Olivier Thibault is a lead author in many of these, but changes in the voltage gated calcium entry, seems to increase with age. So that's an activity dependent function. So then again, it can easily drive a lot of these other oxidative stress, like the mitochondrial, metabolism issues. And so we certainly see that. But these mice don't develop, or at least, you're not seeing the AD pathology
Yeah.
as much. What we are doing increasingly is looking more at neurons derived from Alzheimer's patients.
Hmm.
So both sporadic and the familial form, right? Because the mouse models, you can only really look at familial because you need those gene mutations to drive the phenotype. Where that actually represents a very, very small portion of total Alzheimer's patients. So if we want to see, like, is everything we ever learned in a mouse just specific to the familial case, which, less than 1% of all Alzheimer's patients? Or does it also apply? Do we see it in the sporadic Alzheimer's model? So for that, we really needed to turn towards the, iPSC, or the inducible pluripotent stem cell derived neurons. And the reason why I'm still very enthusiastic about this topic is we see it in those sporadic cases as well. This aberrant calcium handling, certainly leading into mitochondrial bioenergetic oxidative stress pathways. So I'm feeling much better about the mouse model, at least the data or the findings that we were interpreting from, and the human neurons as well.
Okay, so you take fibroblasts, convert them to pluripotent stem cells,
Mhm.
then differentiate them into neurons, and you're looking at calcium elevation in response to stimuli that induce
Mhm.
release from the endoplasmic reticulum, and even in the patients where they don't have a genetic cause, or you see this abnormality. So that suggests some common, it isn't this peculiar to the
Exactly.
familiar cases. Yeah.
Exactly. And, I'm well aware of, the manipulations that we're, introducing is, taking a skin cell, converting it back to basically, a pluripotent stem cell, and then bringing it back up to a neuron. So a lot of the epigenetic factors have been removed. But something is still maintaining this pathogenic phenotype. And we're actually looking at the mitochondrial genes and proteins to see, if those are maintained through this process. Could that, we know there's the free radicals, even in these sporadic Alzheimer's neurons, much higher free radical generations, superoxides. What is that oxidizing, such as the ryanodine receptor and the IP3 receptor, that can alter its properties, make it leaky, push out a lot more calcium? So we're curious if that's the pathway that is generating this calcium phenotype in sporadic neurons after we've done all of these, pretty deep manipulations to it.
Yeah, so the skin fibroblast, that kind of begs the question of, well, at least for the gene mutation, the presenilin 1, for example, is there any, do do people with those mutations have any problem with their skin cells? I don't know that much actually about calcium's role. I know it does play a role in like wound healing and, well, plays a role in almost everything. But
Mhm.
do you know of any evidence for any skin abnormalities?
I do not. But what's interesting is, like, we'll run some of these studies on the fibroblasts and the iPSCs to see what their phenotype looks like, and we don't see any difference in between the Alzheimer's and the,
Oh,
control patients' calcium responses in the fibroblasts.
with the
And
sporadic.
yeah, in the sporadic or familial, for that matter.
Oh, really?
And we'll screen for certain, and we'll do, look for gene expression of certain calcium channels, and we're not seeing differences there. So it seems to be, and I said we haven't done as deep and thorough a dive into this, but, once it's converted to a neuron, now we're expressing calcium channels that aren't in the fibroblasts. Now we're seeing a phenotype that's, not when it wasn't different in the fibroblasts, we're now seeing differences once they're converted to neurons.
I see. I
So
see.
that's thinking, well, if this is mimicking or, mirroring what's happening in, in an intact person in their actual functioning skin, those cellular processes don't seem to be different.
Okay, and then I guess specific kinds of experiments or studies you're doing now, and then maybe you can talk about, the implications or what you found in terms of drugs or other manipulations that can counteract
Sure.
these adverse effects. So maybe first, like, what's your latest thing without giving away something that's unpublished?
Oh, sure. Yeah, no, this is
But you
usually
don't you don't have me to compete with anymore since I'm
I
retired.
actually found you a big source of help and validations, so we were kind of in parallel or,
Yeah.
similar paths. So knowing that you are finding similar things was actually, I found it very gratifying. So again, thank you for all your contributions to the field. Um, so yeah, so what we've done a whole series of studies on this, as a matter of fact. So I'm happy to talk about it. So there's, a series of existing compounds and drugs out there. One is called dantrolene, that normalizes, or it affects that ryanodine receptor, that large calcium channel on the endoplasmic reticulum. And it's a well-known compound. Every anesthesiologist has it in their toolkit,
Huh.
because if they happen to come across a patient with a mutation in one of the subtypes of the ryanodine receptor, the R1, which is found in skeletal muscle, they have a certain mutation in that ryanodine receptor type 1, they go under anesthesia, they get something called malignant hyperthermia, which is basically it's caused by this excess calcium release from the ryanodine receptor in your muscle cells, and your core body temperature goes way up, and it can be, life-threatening. So you slam in dantrolene, you tap down that calcium signaling, and
Is that because the mitochondria become uncoupled? What's generating the heat?
So it's basically, when you have a shivering response to
Ah,
warm up, it's basically doing that. It's creating this very rapid muscle
oh, I
contraction.
see.
So
I see.
it's
Okay.
but after that, I'm sure there's a much better cellular pathophysiology going on that I'm not familiar with that certainly involves mitochondria, no doubt. But it's basically that thermo response to try and warm your body from shivering.
I see.
But now it's gone horribly awry
Wow.
because the coupling between the ryanodine receptor,
So,
um,
so you're shivering when it's not cold.
essentially, I said like a good anesthesiologist would be correcting me right now. But that's essentially, I think what's happening.
Okay.
And so that goes away. But dantrolene will also bind to the other ryanodine receptors. The one we're seeing the most, or we're implicating the most, is the ryanodine receptor type 2, for a couple of reasons. One, it's in high concentrations in these memory encoding regions in the brain, so cortex, hippocampus, amygdala, other regions. We also know work done by like Wayne Chen and Andy Marks that it's aberrantly oxidized and phosphorylated in the brains of Alzheimer's disease patients. So it makes it very leaky and it exposes this dantrolene binding site that normally is closed under, when it's in its normal configuration. And we're finding if we can wash in dantrolene or treat mice with a more soluble form of it, we use something called Ryanodex, we can normalize this calcium signaling only in the Alzheimer's mouse, and only in the neurons were generated from Alzheimer's patients. This
Which mice?
so we've looked at it in the triple transgenic PS1 APP, TGCRND8s. What other ones? Down syndrome mice. We've got a whole parallel project going on with Down syndrome. So even in our Down syndrome mice, we're seeing this.
Huh.
Um,
Interesting.
so
So
right, so
were you in Franklin
Franklin Furley's lab is where,
when
yeah,
they were making the 3-XTGA?
yes.
That turned out to be a really widely used model.
No,
It, uh,
absolutely.
yeah, and it was interesting. In our colonies, over generations, the development of the pathology kind of slowed down, but that was okay in a way because then we could look at uh, interventions in a little better way. Um,
Exactly.
so
Yeah.
you, so in these triple mutant mice, we sent Frank some breeding pairs of our
Mhm.
presenilin 1 mutant mice, and
I
he
remember.
he used this really cool method where he got a female pregnant, took out like the 1 cell or 2 cell stage embryo, and then injected with mutant human amyloid precursor protein, mutant human tau.
hmm
And then that one mouse grew up with all three mutations. So it was a kind of a cool way to make mouse quickly. And but they get amyloid accumulating plaque-like deposits, learning and memory problems, and phospho-tau pathology. We didn't see we couldn't really detect any clear death of neurons, but there's clearly synaptic dysfunction. So you gave this Ryanodex to those mice?
Yes. So we
Okay.
used PS1 APP mice and triple transgenic mice. Again, I always like to use at least more than one AD mouse model for a lot of reasons. They're different promoters, different genes. Who knows if what you're studying is just an epiphenomenon of a
Yeah.
transgene promoter or an actual pathogenic mechanism. So I like to use two models with different promoters, different genes, different, just to make sure I'm interpreting it correctly. So we did in the, PS1 APP mutant mice and the triple transgenics, and we treated these mice for just 1 month, and we treated them either at a pre-symptomatic stage or later when we knew that plaques were already forming and the hyperphosphorylated tau was there. And it was amazing. We did those same calcium imaging studies I was describing before, where we could take out, generate, brain tissue slices, record electrophysiologically. The calcium signals were completely brought back down to normal in the Alzheimer's mice. But what I was actually most excited about was the same treatment in the control mice had no effect whatsoever. right? So it was only taking this abnormal calcium response and bringing it back to where it should be. But if the receptors or the channels were already operating normally, it had no effect, right? And so it wasn't acting like your traditional agonist or channel blocker where everything would go down. It was just taking abnormal and bringing it to normal. And if you were normal, you stayed exactly where you were, like
And
essentially no effect.
did you look at their learning and memory?
So we didn't do behavior, but other groups had. And I'm going to remember their names in just a second. But their learning and memory was also improved
Huh.
in the Alzheimer's mice. So like, you still saw age-related deficits, but you're now operating at the same level as the control mice. We looked at amyloid and tau. It was reduced the amyloid deposits by about 50%, both soluble and insoluble. And then the hyperphosphorylated tau went down like 90%, like it almost completely reversed the tau, which again, we were very excited about. And then it had a whole bunch of other, like looking at synaptic plasticity and other electrophysiological markers, brought back down to normal, that spontaneous vesicle release and the excitatory inhibitory imbalance brought back to normal. And then we've since done a whole series of much more detailed, cellular pathophysiology, looking at mitochondria, looking at lysosome autophagosome function. It was really kind of mind-blowing how widespread this aberrant calcium signaling, like what its influence was
So
on.
it's improving if you inhibit the ryanodine receptors, it's improving autophagy or preventing
Exactly.
accumulation of junk.
Exactly. We were finding that in both the human and the mouse models, the lysosomes, right? So they're the little Pac-Man that are running here. Oh no, it's they're digesting the autophagosomes they're running around clearing up all the unwanted garbage bits. The lysosomes weren't functioning properly because their pH was too basic.
So this is like Randy Nixon's
Exactly.
finding. Yeah, that's
Right. So
interesting.
yeah, I turned to Randy when we were first, observing changes in lysosome. It's one of the proton pumps. We're seeing a significant reduction in the expression of these proton pumps in the Alzheimer's mice and in the human neurons. We're like, what? And then if we treated them with dantrolene or Ryanodex, the levels went back up. And we're like, what the heck is going on here? Because I'm not understanding this connection. So Randy's the first person I called to try and help me figure this out.
He had some evidence that there's a role for calcium in that, right?
Yes.
Yeah.
And it's having to do with the tracking of the vacuolar ATPase, the proton pumps that are on the lysosomes that are bringing in protons to keep them acidified.
Yeah.
So there's a trafficking issue. So they're not getting inserted properly, which is just what we were seeing. And so with that, they can't digest the autophagosomes, so they're accumulating. So we're seeing an increase in autophagosomes, an inability of these lysosomes to digest them. And then if we normalize the calcium, all of this is restored back to a normal, say garbage disposal waste system in the cell. So we were seeing with it accumulation of phospho-tau, right? Because these autophagosomes aren't able to, or, be digested. And so this phospho-tau is accumulating. We're also seeing increases in A beta 42, the pathogenic form. So all of these things seem to be linked.
And is the Ryanodex approved for any clinical indications? Has it been used in humans much?
Yes, so it is used still in that anesthesiologist toolkit. So I believe it's because it's more soluble than dantrolene. So it can get into the bloodstream faster. It can be, you can
Oh,
use
okay.
less. Dantrolene is normally pretty viscous and doesn't go into solution well. So Ryanodex, which is a nanocrystal formulation of dantrolene is used for that. I believe it's also for some other, not malignant hyperthermia, but another hyperthermic condition it can be used for. After
Okay,
that,
so it could be. So what's going on? Is there going to be a clinical trial? Are you getting together with some of the uh, neurology people up in Chicago and do a
yeah,
clinical
so
trial?
we've certainly talked about that. And we were actually trying to develop better RYR2 targeted drugs, because one of the issues with dantrolene and those compounds is it's really meant for, and its highest affinities for RYR1, the one in the skeletal muscle.
I see.
So you really would rather have a higher affinity in targeting the RYR2, so the more brain specific one, and have it work as this negative allosteric modulator, right? You don't want a channel blocker. You want that phenomenon where you take an abnormal response, bring it to normal, but it's not working on the normally configured ryanodine receptor. So we were building compounds like that.
But nevertheless, you saw some pretty striking effects with the Ryanodex in the mouse models of Alzheimer's.
Oh, yes, yes. And said, and in the human neurons derived from these patients too.
Yeah. So why wait to get the RYR2?
Exactly. So we did start going down this path. And the patents for Ryanodex were somewhat prohibitive in allowing us to move forward with clinical trials. And then also it's a question of we needed better blood-brain barrier penetration. So there's groups looking at nasal
Oh, I see.
nasal contribution. So there's some other groups already starting with that. So hopefully, like I'm just interested in and happy to demonstrate sort of the mechanisms of how this is going on. Like should we be putting dantrolene in the drinking water? Like, there's maybe an argument for that.
They'll probably replace fluoride with dantrolene.
Yeah,
Then you won't have any teeth, but you won't have dementia.
Exactly. You'll be around for a very long time without any teeth. So.
What else? So you focused on that. Are there any other manipulations that you've found of calcium regulation that are beneficial?
Yeah, so one of the actually, it's a paper that just came out that my student, Wacy Galagos, and postdoc Sarah Mustale-Kalimi just put out looking at mitochondria. And so we've been very focused recently on mitochondrial dysfunctions. And we know that a lot of this excess calcium being released from the endoplasmic reticulum is being taken up by the mitochondria. And now that the calcium influx channels in the mitochondria have been identified, so the VDAC in the outer membrane and the MCU on the inner mitochondrial membrane, we can see, and with, for example, my collaborator Israel Seckler shares with us genetically encoded calcium indicators that are targeted just to the mitochondrial matrix. So we can measure and see how much calcium is inside the mitochondria. Yeah. And we know now that in our sporadic and our familial Alzheimer's patients, like the neurons we've generated from them, the resting mitochondrial calcium levels are higher. Just at rest, they're spitting out more free radicals. The membrane potential is more depolarized. This is going to affect like electron transport chain and bioenergetic properties. So we're doing a much deeper dive into all of that. And then, consistently with everything else that we've been looking at, and again, this is actually quite a few groups. I don't want to leave anyone out, but if we normalize that calcium with the dantrolene or the Ryanodex, all that mitochondrial function goes back to normal. Right. So we think this is a major driver for the, cellular bioenergetic problems. Once your free radicals are you're just spitting out too many, it's, when I give a talk to more of a lay audience, I bring up that commercial of, I forget what insurance company it is, but like, the mayhem guy, right? So these free radicals are just creating mayhem by aberrantly oxidizing all of these proteins and things that shouldn't be, and creating problems. One of them is probably the ryanodine receptor. So this is, I think, where we're getting this feed forward spin. There's a whole multitude of things that can be driving mitochondrial stress and oxidative free radicals. Doesn't have to be calcium. There's a long list there. But once you start oxidizing that ryanodine receptor, particularly the RYR2, and it starts leaking out more calcium, well, now you've basically like open Pandora's box in terms of what could possibly go wrong in terms of, synaptic structure, synaptic function, dysfunction. That whole lysosome autophagosome cascade can now go awry. So it's it's not so much, do I think the ryanodine receptor or calcium is the, starting point for all thing else, for all the Alzheimer's pathogenesis? Not necessarily, but once you invoke that, I think that's the engine that's driving this feed forward cascade. And if you can stop that engine, or at least get it functioning normally again, everything it's coupled to also is restored back to normal. And that's been our findings so far.
There seemed to be, as you know, we had a lot of interest in, how can one reduce their risk for developing Alzheimer's through like lifestyle modifications,
Yeah.
exercise,
Mm-hmm.
intermittent fasting, intellectual challenges. It
Absolutely.
would be interesting to see how any of those
Mhm.
good lifestyle factors impact the endoplasmic reticulum calcium
Absolutely.
regulation.
Yeah, that is a great question. I'm again a colleague here, Judy Patashkin, who's very those are the sorts of questions she really focuses on, not so much the neurophysiology, but the mechanisms by which healthy lifestyle changes and interventions are impinging on the disease process. So we're trying to pair up. She's very interested in glucose metabolism,
Yeah.
insulin handling, things like that. So we've got some preliminary studies definitely supporting a role for all of that.
Okay, so you're doing like uh, the kinds of imaging, calcium imaging and recording from neurons from animals that have been with different, whatever, different states of glucose regulation or
Mhm.
insulin resistance, that kind of thing.
Well, this is actually more in the human neurons, and we're looking at gene expression patterns of several of these key regulators. So glucose transporters,
Oh, I see.
insulin receptors. We haven't done too many manipulations on them yet. We're just getting a baseline
Oh,
reading.
oh, you're looking at those downstream of the ER
Yes.
rather than looking at how, for example, exercise or fasting affect the ER.
Right. So we would like to do that. So again, it
Yeah.
it is on the more time, more
Yeah.
funding, more bandwidth. But I
Yeah.
think those questions are really interesting because we all know it's helpful, but what we don't really know is like the precise cellular mechanisms by
Right.
which it's
Yeah.
it's working. Another one that I find really interesting, and again, it's highly correlated with being, reducing your risk of Alzheimer's or dementia is, social interactions.
Yeah.
And I find that fascinating. And there's specific region of the hippocampus, the CA2, and that CA2 connection to CA1 is thought to be very important in sort of in driving and regulating those sorts of social interactions. And we have, again, imaging data, and this is with Bill Frost, where we can image an entire hippocampal slice and trigger different pathways in it and look at the strength or the voltage changes across it. And we're seeing deficits in that CA2 pathway. And we never knew what to make of that. And so, this is years later, all this, data and epidemiological findings suggesting that, having a socially enriched lifestyle is very protective. We're like, we need to go back and revisit that pathway. And then, is this also coupled with, again, healthy lifestyle interventions, things like that. So that's kind of where we're starting. We said I actually have a new graduate student in the lab. He'll partially be looking at this. So Danny Leserve. So we'll see what comes out of her thesis. But those are some of the things we'll be looking at, is that's that pathway.
That, yeah, so you have this row of the pyramidal cells are, kind of semicircle, and you've got CA1 neurons, CA2, CA3. And the CA2 neurons have a lot of interesting features. One is they seem to be more resistant to excitotoxicity.
Mhm.
So if you induce epileptic seizures in an animal with a glutamate receptor overactivator, and then you look at which neurons degenerate, CA3 and CA1 are vulnerable, CA2 isn't. So they have, I mean, that kind of very indirectly, but it kind of suggests some difference in calcium
Yeah.
regulation, or maybe have better calcium buffering, or, well, I guess it's just very complicated because the glutamate receptors, uh, and whether the AMPA or the NMDA, the calcium channels, are differentially expressed. But that doesn't seem to, for example, the dentate granule neurons are very resistant to
Yeah.
seizures, but they have high levels of NMDA receptors.
But also high levels of calcium buffers.
Yeah, yeah. Yeah, so maybe there's something different about the ER and the CA2 neurons. That would be interesting.
Yeah, no, so we're excited to get started on this. So
Yeah,
that's some of the newer directions we're going into.
yeah, good. Um, what else? Anything else you'd like to talk about?
Yeah, so it's actually an exciting project that I love to talk about. And again, it's very much paralleling what we're doing in Alzheimer's disease, but looking at Down syndrome, right? And so you think it's the other end of a developmental intellectual disability. Um, but what, I have come to learn recently is now that people with Down syndrome are living longer, and in large part, they can correct a lot of the congenital, say, heart defects and things like that. These people are living much longer. And what they're finding is that their likelihood to develop Alzheimer's disease is markedly higher than in the general
Yeah,
population. So in their 30s and 40s, a large percentage of them are now developing Alzheimer's disease.
yeah,
So you have an intellectual disability, and now you're developing dementia. And what I find, well, highly motivating is that, now that, many people, either at birth or even in utero, that this person will have Down syndrome. You now know that they will also very likely develop Alzheimer's disease. So you now have an opportunity for intervention on day one.
yeah,
And maybe along the way, you can also learn what are some of the key driving factors for developing Alzheimer's disease in the general population. So we're really studying what are some of the common early upstream mechanisms shared between Alzheimer's and Down syndrome. And if you are able to, introduce some sort of intervention like normalizing calcium, can you prevent or reduce or significantly delay this Alzheimer's disease onset? And I think this is a critical question. One, I mean, not to get on my moral high horse, but like if you know this population is going to develop dementia, like you must do something about this, right?
yeah,
This is a tragedy that can largely either be prevented or I think needs more attention. And you can also learn a lot of very valuable information. So we're working again with mouse models of Down syndrome, as well as the human neurons derived from people with Alzheimer's disease. And this is with groups such as Steven Ginsburg at NYU. He and I have a grant together to study this. And we're looking also a lot of like their end does a lot of the transcriptomics and proteomics from the mouse and the human populations. And a lot of these differentially expressed gene pathways are focused around mitochondria, synaptic, and calcium handling pathways. And we see it very, similar ones between Alzheimer's and Down syndrome. And so we started with, okay, we see these differentially expressed gene pathways. Interesting, but I guess I'm more interested in what the function is. So, are these functions coupled to these gene pathways also altered? So we're looking at mitochondria, calcium, like all of these things, and we're seeing very profound differences in the Down syndrome neurons. So their ER calcium is much greater. There are some differences, such as just resting calcium seems to be higher, but this is also seems to be mediated through the ryanodine receptor. We're seeing that same mitochondrial free radical oxidative stress pathway kicked in. We're seeing changes in the lysosome, both functionality and proton pumps. So a lot of these cellular pathogenic mechanisms are in play in the Down syndrome neurons, both in the mice and in the humans. So we're wondering, okay, if we tap into that, can we prevent or reduce their later conversion to dementia?
I've thought quite a bit about the familial Alzheimer's disease, for example, down in South America.
Mhm.
Colombia, there's a bunch of families that have the same
Yeah.
mutation, and it is possible, theoretically, it's possible to eliminate those from in
Hmm.
one generation because, um, so if you have a parent that's developing dementia in their 30s or 40s and you're one of their kids, you and that you have a 50% chance of having presenilin mutation. So you could get genotype to see whether you inherited the mutation.
Right.
If you didn't, that's good news. And then you don't have to worry about passing the mutant gene on to if you're going to get married and have kids. But there is also a way that even if you have the mutation and want to have kids, you can ensure that the kid does not have the mutation because you do in vitro fertilization and embryo selection. And you could select like this is like 2, 3, 4 stage embryo that doesn't have the mutation, and then put that in the uterus of the mom. And but there's ethical considerations because some people think that's whatever playing God or something like that.
Mhm.
And Down syndrome too. I mean, I guess I should know this, but there is screening, right, to see
Yes.
whether, uh,
Mhm.
uh,
I
yeah,
believe it's second trimester. I'm honestly forgetting when the screening, and there's a couple of different ways to do it. Um, so there's screening, right? And then it becomes a moral, a personal choice,
yeah, yeah,
what you do. But, I don't know, right? And and IVF and embryo selection, it's not a trivial process. So
no,
I think it's a much bigger questions and much bigger moral implications for people to decide, and it's
yeah,
definitely not
yeah,
our to
yeah.
discuss.
And I don't know, is it possible to correct the gene or silence the mutant gene in all cells in the body and brain
Right.
of a person? That's still a long ways off.
Right. Well, I mean, some of the CRISPR,
The CRISPR stuff.
gene modifications are being used for some, genetic diseases. So I think it's perhaps a little early stage for something like a mutant presenilin or familial
Yeah,
Alzheimer's disease. And that is not my area of expertise. I look to my colleague Bob Marr for those sorts of answers. But it's certainly, being able to manipulate gene expression is increasingly being used now that some of the earlier complications seem to have been resolved.
yeah. Okay. Well, thanks, Beth, for taking the time. I hope you have a great rest of the summer.
Yeah, it's been wonderful chatting with you again. Glad to see you're still up to exciting things. So thank you for that.
Okay. Bye.
All right, take care.